Thank you so much, Mike, for having me here. It's a pleasure to be a part of VenomWednesday and give a talk. Uh, Today, my talk is going to be about a part of my PhD thesis, where we are trying to understand how different venoms are and what are all the different factors that are probably influencing this variation. And most importantly, how this affects the snakebite therapy. So I'm a graduate student. I'm in my final year trying to finish my thesis in the Center for Ecological Sciences. I'm working with Professor Karthik Sunagar in Indian Institute of Science, India. I come from India, which is a culturally very diverse country, but it also biodiversity rich country. We have very different climatic and biogeographic zones. And this biogeographic diversity actually also harbors a huge biological diversity. And we have over 300 or 350 species of snakes in the country. But less than 1/3 of them are actually capable of inflicting life-threatening bites to people that can actually kill people. And even among them, a major proportion of bites are attributed to two of these snakes, the Russell's viper and the common cobra. But these snakes are also very interesting from ecological and evolutionary perspective. Their venom is very interesting in terms of their composition, in terms of their function. But we very poorly understand about these different factors that influence their snake venom phenotypes. So the major factors that people have attributed to venom variation are environmental factors, including abiotic and biotic factors, such as temperature and pressure and elevation, and also biotic factors, most importantly, the diet of the snake. But venom variation has been shown in these species from some populations. But in my PhD, we also tried looking at several other populations that have not been characterized before, and also wild populations of these snakes that are naturally consuming their diet in the wild. So we looked at these different populations and we found remarkable diversity in terms of their protein composition, in terms of the relative abundance of different toxins present in the different populations of these two snakes, the common cobra and the Russell's viper. But we were also interested in understanding how venoms vary across the developmental stages of a snake, that is the ontogeny, and also how the venoms vary between different sexes. Why should the venoms vary in these different stages? Because venom is an adaptive trait and it is related to the functional, the function of the snake, that is carrying out the predation or defending itself from predators. So the hypothesis here is that the venoms of these two snakes, which are distributed all across the country, found in very different agroclimatic regions, might also vary ontogenetically because as a baby, they might be feeding on different type of prey, and as they grow older, they might be feeding on different types of prey. And they might also be defending themselves from different types of predators as they grow from young to older snake. So to look at this variation, we collected venoms of young individuals and adults from a particular region in India. So this was done to avoid the geographic variation, which we already know. So the location was kept constant, and these adult mothers were housed and the babies were born in captivity and their venoms were collected periodically every 3 months. So we classified them as neonates, as snakes that are less than 30 days old, and juveniles anywhere from 1 month to 12 months old, and adults are anything that are over 2 years old that is capable of sexual reproduction. So just to give you a visual idea, this is how small the babies look when they emerge. And we wanted to look at how potent is their venom. So when we did different proteomic and functional studies, we found that the venoms of these snakes vary between the adults and the younger stages. The adults express a lot of higher molecular weight toxins, which are found in very less abundance or almost absent in the in the younger individuals. So these are the venoms of the neonates, these are venoms of juveniles from 3 months, and these are venoms from 6 months, and so on. And in all of these, when we compared them with their respective adult mother samples, we were able to see that there is a stark shift from a low molecular weight abundant toxin to a high molecular weight abundant toxin in case of the adults. But the interesting thing that we also saw is that in the case of the juveniles, at around 9 months, they started shifting to an adult-like phenotype. We also did mass spectrometry to look at the composition of these individual toxins that are actually shifting from the young to the adults. And we found that the venoms for adults are actually more complex, and they produce different types of toxins that are not actually found in the venoms of the younger individuals. They also start expressing high molecular weight toxins such as SVMP, which is not found in the babies. So we wanted to correlate this with their actual toxicity to see how this diversity actually helps in the toxicity in capturing different prey organisms. So we tested the venoms against mammals, which is one of the major target prey organisms for the Russell's viper. And we actually found that the venoms of both the adult and the juvenile Russell's viper were more or less similar, and they were actually toxic. That is, very less amount of venom is enough to kill the mouse. But what was more interesting was that the venoms of the babies, that is, any Russell's viper that were less than 30 days old, were actually twice more potent than the adults. And the toxicity was so high that the dosage was actually very similar to that of the toxicity of a neurotoxic common crate, which is one of the most potent snakes in India. So the babies are actually more potent. But is this just against mammals? We wanted to also test against other prey organisms because we have observed during our study that they have this behavioral trait where the neonates or the babies move their tails in such a way that it looks like a worm. And they specifically do this behavior only in front of smaller animals like frogs, and they don't exhibit this behavior in front of mammals or big rodents. So we tested the venoms of these younger and adult individuals against common house geckos or lizards. And we found that the babies, that is the neonates and juveniles, are actually toxic against geckos as well. But interestingly, the venoms of the adults did not exhibit any toxicity even up to 20 milligram per kilogram. And that was very interesting because the diversity of the venom was very high in the Russell's viper adults and not in the babies. But in terms of toxicity, the babies were actually toxic against both the lizards as well as mammals. This is possibly happening probably because the adults require more diverse venom to bring down larger prey, such as rodents, which have a larger body mass, and they need different mechanisms to quickly immobilize that prey. Whereas the babies are using a very potent cocktail made of just few components to bring down smaller prey organisms. And it is also toxic against laboratory mouse and other prey organisms such as geckos. And we also think that this kind of increased toxicity in younger individuals might also be a way to cope up for the very little amounts of venoms that they produce when they're babies. But what about the snake that is also occupying the same biogeographic zones found in the same region? What about the common cobra venoms? Does that also experience a similar shift? If we think about their availability of prey and so on and so forth, we would assume that a similar shift might be experienced by these snakes as well. But that's not what we observe. And we saw that the adult and the baby venoms were more or less very similar. Proteomically, we also looked at the mass spectrometry, even though we did find few components additionally in the baby venoms. The most important medically relevant component found in the venoms of common cobras, which is the neurotoxins, were found in more or less similar amounts and also in similar diversity. Why I say diversity? Because we also looked at them functionally, where we look at the binding between the nicotine acetylcholine receptors and the venom toxins, the three-finger toxins, which are the major components of the common cobra venom. We found that they bind very well towards different prey receptors such as frogs, rats, and lizards, and they bind poorly against some of the organisms that are known to be resistant to these venoms, such as mongooses. And that pattern was more or less similar for both the babies, such as juveniles, as well as the adult snakes of common cobra. We also tested toxicity in mammals as well as in geckos, and we found that the toxicity was also more or less similar for both the adult and the baby common cobras. So this made us think that it is possible that the common cobra and the Russell's viper, both of them are extremely potent towards mammals. But the common cobra is also potent towards geckos, whereas the adult Russell's vipers are not potent towards lizards or geckos. This might possibly hint that the common cobras, which have a very diverse three-finger-toxin composition, the neurotoxins, which can target very different types of prey receptors, do not experience any shift in their venoms, and also probably this enables them to be a generalist, that is, they can feed on very different type of prey organisms. Whereas the Russell's viper, especially the adults, have a venom that is probably more suited and evolved in such a way that it targets only mammalian prey. We also wanted to confirm if this would be the case for different populations. So we took venoms from different populations all across the country. And when we looked at the common cobra venoms in mammals and geckos, we saw that the venoms were more or less similarly toxic against both the mammals and geckos. There was a lot of intraspecific variation between different populations, but overall, if you see, both of them do have toxicity against both geckos and mammals. But in the case of Russell's viper, we were able to see that the Russell's viper were extremely potent towards mammals. That is, if you see, this is 1 mg per kg, that is very less amount of venom is required to kill mouse. But in terms of the dosage for the Russell's viper populations, some of them are as high as 20 or more, or in case of the Karnataka, which I told earlier, the geckos don't die at all, even up to 20, which shows that the Russell's viper venoms are probably not targeted towards geckos even across different populations in the country. So I'll finally finish with saying about how these variations actually affect the snakebite treatment, because most of the studies are done in the light of understanding how venom variation affects the snakebite therapy, because most of the antivenoms that are prepared in India are prepared by collecting venom from just one population, and these are also from just adult snakes. We have previously showed that this antivenom does not work in a lot of places which is far away from where the venoms are collected for making antivenoms, and also against a lot of species that are not used in the antivenom process. But what about the ontogenetic shift? What we saw here was interesting, that the antivenoms did not work for the cobra or the Russell's viper, either for the babies or for the adults. The antivenom just does not work against this population at all. Even though here it is very difficult for us to say whether ontogeny is a problem for antivenom, the bigger problem here is the geographic variation, because the population that we have tested here is Karnataka, which is again a different population than where the venoms are collected for making antivenoms. This antivenom just does not work for any of the samples that we use in the study. So to summarize, the Russell's viper venom does exhibit an ontogenetic shift across developmental stages, which is not seen in Naja. These Russell's viper venoms have also evolved to express highly toxic low molecular toxins in their juvenile and neonate stages to compensate for the lower amounts of venom that they produce. Both the adult and juvenile Naja exhibit certain prey specificity to different prey receptors, but there is no shift in terms of ontogeny. And we also observed that geographic variation plays a major role in determining the antivenom efficacy than the ontogeny, or at least we could not differentiate the two. And this points out that we should probably look at making region-specific antivenoms to source venom from different populations where antivenoms are working or not working, and also look deeply to understand whether these other factors such as prey specificity and ontogenetic variation really have an effect on antivenom efficacy other than just geographic variation. A lot of people really helped me in many of this work. I would like to express my gratitude to my professor, who has been with me throughout the process of my PhD, and also all the forest departments who have given us permission to work on these venoms, because it's not like these snakes are protected by law, so not anyone can work on these snakes. So we took special permissions to work on them, and also our collaborators, the organizer Mike, for giving me this opportunity, my lab mates who are also part of these studies, and also different people who have contributed to the photographs that I've used. And also these photographs gave us very valuable information about the ecology of this species. So my thanks to them, and also to all the funding agencies. Thank you so much for listening.
Thank you very much, Senji. Impressive, really impressive to see that the elapids and the vipers are totally different, at least for these two species, in their ontogenetic shift. I think we have to see if the question were maybe already answered after they were written. So the first one is from Damien. Great talk. Is the difference in snake venom between juvenile and adults from an increase in the production of certain toxin families quantitatively and others not, or simply a different toxin family profile in the number of sequences per toxin family qualitatively in your analysis? You can see the question in the chat
Yes.
also, if it maybe was too long.
Yeah. Okay, so there is a difference in terms of quantitative expression. Definitely, the relative abundances of toxins are definitely different. But from mass spec, from shotgun proteomics, we were not really able to capture the sequence differences between these two toxins, even though we strongly believe that the PLA2s, which is the common protein that we see in both the adults and the babies, are probably functionally different, because I did not present those data, but we also did functional testing, and we see that the PLA2s that we find in babies have hemotoxic activity. They are also catalytically active, whereas the PLA2s that we find in the adults are not catalytically active. They might have other non-catalytic toxic functions that we don't know of, but at least in terms of catalytic activity, they are different. So they are definitely probably different in terms of sequence as well. But from the studies that we have done, we were able to capture the quantitative differences. And also the number of sequences, I think the quantitative aspect would mean that the number of sequences per toxin in transcriptomics is obviously different, that they are like one of them is producing obviously more number of transcripts and hence more protein than the other. There's also one more follow-up. Great talk for the, thank you so much. For the local antivenom production, both juvenile and adult venoms is applied or only, yeah, it's only adult. So far, it is just from one population and just from adults, because no one even thinks that the babies can have such potent venom. Are there regions where the local variation is so different that the available antivenom can display severe effects, like the Indian antivenom in Sri Lanka? The adverse effects of antivenom are probably because of how antivenom is made itself. It is from a horse and it's very crude and it's not very purified. So irrespective of whether there is variation or not, I think the adverse effect would still be there. But the effects are amplified, because if you're using an antivenom and it is not serving any purpose and just keep increasing the dose thinking that it is not working, then it would just make the adverse effect worse. So I would think that would probably be how the variation could affect the adverse effects of the antivenom.
Thank you very much, Senji, for all these answers and the audience for the questions. Are there further questions? Else, I have a short one. I know from different regions that there are then often the legend that juveniles are way more venomous or toxic or lethal than the adult snake. Is it the same in India? So that it's often said and nobody tested it before? Or how did you came up with the study?
Yeah, this was the first study from the Indian venoms that we have tested on Indian populations. I think the legend keeps varying. Some people believe that the babies are just too toxic because they release all the venom and they don't know how to regulate the venom injection and all that. But I think from our study, it's very clear that the venom as a characteristic itself, it's more toxic than the adults. But this was just one population that we have studied. In the lab, we are also studying other populations to see if this holds true for others. But yeah, this is not widely known across different populations.
Okay, then I don't see
one more question.
One more question.
So first of all, great work and great talk. I was curious because you mentioned in a couple of moments that potential predator pressure could also be the driving factor of the variation in venom composition. Do you have some plans to conduct experiments or the assumptions in which way and which kind of predators, how actually predators are changing throughout the ontogeny as well.
Yes. So I said that in terms of those factors affecting venom variation in general, not just in ontogeny. We do have studies where we are trying to look at that. Unfortunately, we can't directly test this on the snakes, as in subject them to different pressure conditions. So we have been collecting venoms from different regions which have different temperature and altitude and pressure. So these are the abiotic factors that we're considering. But we don't think that these might directly have an effect. They might more kind of have an indirect effect where these abiotic factors affect the prey density, and hence the venom also has to change in association with these differences in the abiotic factors, and ends the difference in the prey. But yeah, we are trying to do some of these tests as well.
Great, thanks. Yes, just that. Thank you.
Thank you.
Looking forward to seeing it also published, everything.
Yes, yeah, we have submitted, so I'm just waiting.
Cool.
It gets published soon.
So there's another question in the chat. Did you use UV visible for the quantification aspect of the venom composition? And if so, do you think that all toxins obtain in the same way?
We did the UV one, okay, so I think you're trying to ask if we just did UV visible for the quantitative aspect. We have actually done multiple levels of fractionation. We did hydrophobicity-based fractionation, then RPS-PLC, and then we run an STS-based fractionation, and then we subject them to mass spec. And in mass spec, it's ionization-based. So I do believe that not all toxins will ionize the same way or separate the same way. That is always a downside of mass spectrometry with venom toxins. I think might be able to shed more light on this. So yeah, we have subjected them to different levels of fractionation to try to normalize for some of these inherent differences in the venom toxins. But yeah, there is definitely a possibility of that. But yeah, we have not used just UV visible as such for quantification.